you are the Chief Science Officer of Yuvan Research. So welcome to Modern Healthspan, and thank you so much for joining us today.
My pleasure.
So, Dr. Katcher, so your team recently published a paper on bioarchive, so reversal of biological age in multiple rat organs by young porcine plasma fraction.
Right.
Some updates to your work on E5. So could you talk a little bit about, so what is new in this latest paper? What's kind of the updates in it?
Well, as you detailed in your own podcast, I revealed the secrets of E5, which is basically two things. One is that it's primarily nanoparticles, like exosomes, that are the basis of rejuvenation. And that completely disagrees with what was previously thought by people like the Gun Boys and Saul Valeda, etc., Amy Wagers, who all attributed anti-aging of blood plasma to small molecules, proteins. I never believed that was true for some obvious reasons. For example, any single gene can be mutated, and the lack of that gene or the over secretion of it should produce vast differences in lifespan, and that doesn't happen, except for lamin A. And even that's a question of alternative splicing or alternate splicing. And the second thing is, and I consider this really the most important thing because it makes it a viable option for all of humanity, is that it was porcine plasma, not human plasma. And that leads to two things. One is the fact that porcine plasma is apparently, or rather the nanoparticles, the exosomes, etc., in porcine plasma don't have any immune reaction in rats or in that, for that matter, in human beings. In the one instance that I successfully tried it, which is my own hand. I think you've seen pictures of it, or shall I show you?
Yeah.
I don't know how well you can see this.
Mm-hmm.
This is my this is my untreated hand. This is my untreated. So it's kind of confusing. I got a mirror image, I think.
Right, yeah.
This is my untreated hand, and you can see that it's quite discolored, and the skin is so thin that you can see the scars left from, I don't know, decades of of working.
Right.
And this is a treated hand, and there are no scars visible. The skin is much thicker. There are no wrinkles. I don't know how this camera is not really so great, but my hand is all wrinkled, and this hand is not.
Yeah, looks a lot better. Yeah.
Yes, and if I had a better camera, you could see that there are no wrinkles, and it looks like a young hand. And there was one treatment about 1 year and 5 months ago, May of 2022. Period. One treatment.
Yeah.
1 and 5 months ago, and the effect persists, and it seems to me exactly as good or better than it was originally.
Yes.
So, and it took 4 days to take effect, and there were no ill effects whatsoever. I mean, I took a shower after my my first day, as people do in India,
Mm.
since you get all hot and sweaty,
Hmm.
uh, and that was enough.
Yeah, yeah. And I I kind of want to come back to topical E5 a bit later. Uh,
Sure.
yeah. So exosomes. So we think, or we know they're exosomes. So within the exosome, do we know, because exosomes contain a number of different factors, right?
Correct. Correct.
And so do we know which ones are producing the effect?
At this stage, nobody knows.
Mm.
I suspect it's microRNAs, which are significant in controlling translation.
Mhm.
I suspect it's also RNA binding proteins, which are very much disturbed in the aging process, and RNA processing proteins. Uh,
Mm.
and for instance, in the older you get, in any species we've any mortal species, the more um, uh, alternative splicing we get, so that the proteins that are produced are different
Mm.
in the aged person or animal than they are in the young one. Uh, the alternate splicing, for instance, that causes the premature aging of progeria, uh, is absolutely. I mean, it produces, instead of the normal lamin A protein, it produces something called progerin. And if you look in young people, normal young people, there's zero progerin.
Mm.
But as you age, the amount of progerin in your blood increases. So that old people are actually starting to produce progerin, which to me is just it's not the cause of aging, but it's just a sign of alternate splicing, where instead of splicing is a process whereby the RNA made inside the nucleus, called heterogeneous nuclear RNA, has parts of it cut out, and 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 the bad parts,
Mm.
called introns, are spliced out, and the good parts, called exons, are spliced together.
Mm.
And it turns out that there are various possibilities whereby an intron can be left in the splice, or an exon can be left out of it, or an exon starts at a different place. The start of the messenger RNA that codes for protein starts in a
Mm.
different place. And that is typical of all aging animals. That, plus the presence of circular RNAs.
Mm.
And, and I believe Steve Horvath recently put out a paper talking about lifespan, and he noticed that the major changes were not changes in the methyl groups that inhibit translation of proteins. For example, when you age, your repair enzymes, DNA repair enzymes,
Mm.
decrease, so that the DNA repair decreases. But that's not because you make less of those enzymes. It's because they're alternately spliced so that they don't work as they should, as they
Right.
ought. And
Mm,
I believe
okay.
the secret of aging is modifying those methylation sites, basically reprogramming the genome, because Steve Horvath's work shows that the genome is reprogrammed by E5.
Mm, yeah. So, and
I'm sorry if that's not clear.
no, that is. I mean, I see the splicing. I mean, we talked about the way that with yeah with another researcher about the way that splicing affects the RNA after it has been
Correct.
created.
But
And
what's not realized by most is that intergenic methylation sites, which
mm,
are what mostly change during aging, also affect splicing.
right. Mm,
So gets a little complicated, and I'm really working hard to understand it, to write about it.
yeah. Sleep is the key to your body's rejuvenation and repair process. It controls hunger and weight loss hormones, boosts energy levels, and impacts countless other vital functions. During the holiday season, it's easy to slip from our health routine and have more late nights and eat irregularly. In fact, drinking more than 2 servings of alcohol per day for men and more than one for women can decrease sleep quality by 39.2%, according to a study from Tampere University in Finland. But when the vacation season winds down, it's time to get back on track and focus on healthy eating, exercise, and above all, quality sleep. For my sleep, I take Magnesium Breakthrough by Biooptimizers because it contains all 7 forms of magnesium. I take it every evening and it helps me fall asleep, stay asleep, and wake up refreshed. Visit magnesiumbreakthrough.com/modern and enter code 10 for 10% off your order of Magnesium Breakthrough. Thank you for your support. So at the moment, so we have exosomes, and the exosomes. So it's really interesting that the exosomes work across species, right? So,
Yes.
is it?
So this is a highly conserved process, obviously. I mean,
Yes,
at least it works across all all mammalian genera,
right.
as far as we know. Uh,
Yeah. So,
yeah.
so it's anything special about pigs? I assume we could use other animals as a source.
Well, you know, as you know, I did my research, most of it in India,
Mm,
and to try to get cow blood, for example, was
yeah.
like trying to get your grandmother's blood for for experimentation. So that was very difficult. We tried with water buffalo
Hmm.
blood. So for some reason, water buffalo, even though it's the same genus, Bos, is not sacred to Indians. So they didn't mind, but the water buffalo minded an awful lot.
Mm, yeah.
And we could only get a few cc's out. And we tried it. We found that when we applied our procedure, we got the very strange uh, precipitates, things we didn't understand with the structures, cubical yellow blocks.
Mm,
Uh, so we didn't know what to make of it, and we tried it, and it didn't seem to work at the time, but we didn't really know what we were doing at that point.
right.
Uh, the idea about pigs or cows, specifically cows, is what I wanted to start with, because there are about 2 billion cows on Earth, and you can remove the blood from a cow, significant quantities of it, without injuring the animal, whatever.
Mm,
Now, since cows were out and sheep, goats, etc., are not raised in such enormous quantities and are kind of hard to draw blood from in any case, uh, pigs are raised for one purpose, and that's to be slaughtered. They have no other economic value other than their meat, and perhaps to some extent the leather, to a very small extent the leather. And there are about 1.4 billion pigs slaughtered each year, and they're slaughtered at almost precisely the right age that we would prefer, which is late adolescence, early adulthood. And at that stage, they're relatively large animals, and you can get a significant amount of blood from them. And blood is half plasma, so significant amount of plasma as well. So to me, this seemed to solve the entire problem that, this extension of youth, at least, wouldn't be reserved for the very rich, but could be a worldwide market for almost anyone, as far as cost goes, because because pig blood is basically an industrial byproduct. I mean, it's used to make blood sausages, but there's not that much consumption of blood sausage.
Yeah.
Otherwise, it's used as fertilizer and or just thrown away. I think we were paying $3 a gallon for it back in the U.S. So it seemed to me this solved all of the problems. One, it was guesswork that it would work in rats and humans without any immunological reaction. And that proved to be correct. And it was also guesswork that it was the exosomes that were important. And that proved to be correct. And with 1.4 billion pigs being slaughtered each year, they breed very quickly. There are only about 650,000 pigs in the world, but they slaughter 1.4 billion pigs each year because they keep on reproducing and have large litters. And the only purpose to breeding them at all is for food, and then the blood is just thrown away. And here we've made it into perhaps the most valuable resource on Earth.
Yes. So is E5, when you're narrowing down E5, so E5 is just exosomes. Is that, it's just like pure exosomes, or are there other elements from the plasma left?
There are contaminants, of course. For instance, albumin seems to be a contaminant. But what I'm wondering, and what we don't know yet, and we're doing experiments now to find out, is whether there are other nanoparticles aside from exosomes. There are also what they call ectosomes, which bud from the cell surface. There are exomeres and what they call supermeres, which are even tinier than exomeres, and both are tinier than exosomes. And my purification method was designed to capture all of them. So hedging my bets, as it were. So yeah, we don't know yet, but we're doing experiments to find out.
So exosomes come out of cells. Do we actually know which cells these exosomes came from, or does it matter?
Well, I think that's the point. Exosomes come from pretty much all cells, and the exosomes in blood come from all cells.
Mm.
And different exosomes contain different contents and have different targets. The contents are called cargos in the literature. So exosomes have different cargos, and they have different targets. So our thought was if we take all the exosomes, which could only be found in blood,
Mm.
and inject them in fairly massive amounts, which can't be done with human blood,
Mm-hmm.
uh, that we would have the effects that we had. So, for instance, in the paper that you referred to by Shamagian,
Mm-hmm.
uh, they used exosomes from cardiomyocyte spheroids, and
Mm.
those seem to have a general effect, but it took 4 months of injections, which to a rat would be equivalent to about 10 years in a human. And these results occurred in 4 days. And even with our rats, we saw the effects in less than a week, or
Mm.
approximately a week. So, which even in human terms would only be a couple of months at
Mm.
most. Uh, so we think it's a combination of the variety of nanoparticles plus the amount of nanoparticles that that make all of this possible, and within a limited time period. You don't want to be taking injections for 10 years to see an effect.
No, no. So, I mean, could it be a dose thing? How confident are you on the dose you're using?
um, we're pretty confident because when we have the dose, we had no effect, or a very transient effect that soon disappeared. So I think dose is a very important part of it. And
I,
this is again something that you can't do with human blood unless I hate to think of the ways that one might get such material.
yeah. I mean, there are like placental-based exosomes which you can get, but I guess it's the quantity that would be the problem.
Exactly, exactly.
Right. So,
And the same with those cardiomyocyte spheroids. You'd have to grow them and others because there's no reason to assume that they'll affect all organs. I didn't see any effect on cognitive improvement,
did
for example.
did they test
Although
that?
I
Yeah.
don't recall that they tested them,
Right.
but I know, for instance, that Kai Dongxing Kai at Albert Einstein University in New York uh, used neural stem cell exosomes to to rejuvenate mouse brains. He had to inject them right into the third ventricle of the brain, uh, and the effect wasn't great. It increased lifespan by about 15%, maximum lifespan by about 15%. Uh,
Mm.
but there's no reason to believe that the cardiomyocyte exosomes would have the same effect on neurons as neural stem cell exosomes. So with blood, we get all of it,
Mm.
and we're thinking about exosomes from other bodily fluids as well.
All right.
because just about all bodily fluids contain exosomes, and not necessarily the same.
So, interesting. Okay. So when you looked at the rats, so it rejuvenated all the places that you looked, right? Well, except for the brain. The brain was somewhat rejuvenated, but not as much.
Well, let me say that the hypothalamus of the brain
Well, all right, the
was
hypothalamus.
somewhat rejuvenated because we saw significant cognitive effects. We saw a significant improvement in cognition among the old rats.
Hmm.
Eventually, after training, they got down to the same level as the young rats, while the untreated old rats just kind of didn't improve whatsoever and actually got worse with time.
Hmm.
So we actually saw reverse aging,
Right.
youthening, I called it.
Yes. So you did run an experiment for females only, right? To try and see how long the total lifespan would be.
Correct,
Yeah.
correct.
And
That was the
that,
choice of our PI
right.
in India.
And
Who's
and that
who's
that showed
female.
that showed some improvement for the oldest. Yeah, I mean the oldest rat was.
It showed improvement for all of the rats,
Hmm.
but for the oldest rat, Seema, which
Hmm.
uh, which means boundary or limit in Hindi, uh, she lived to the equivalent of about 150 years in human terms. So a really significant difference. 6 months longer than the
Hmm.
normal lifespan,
Right.
which again, with rats, 2.5 weeks is about 1 year of human life.
Yeah. And you stopped. I mean, it was every 45 days, right? And but then
Yeah.
you stopped at some point. Did you continue?
Yes. Quite honestly, I wasn't in India at the time, and I don't know why
No.
they stopped.
Okay.
I would have liked to have seen it continue,
Uh,
but they stopped. But even so, she just lived and lived and lived.
yeah. Did you have any further thoughts on why it seemed to impact the brain less? You did talk a little bit about it in the paper.
Well, the brain is a different organ than most others in that it has a very selective delivery system from the blood to the brain called the blood-brain barrier. And yeah, I can only think of the blood-brain barrier was the barrier to the
Right.
complete infusion. And I think if perhaps we injected the exosomes into the cerebrospinal fluid, we would get much better results. It's something we're thinking of doing, of course.
Right. Yeah, that would be interesting to see how that works.
Yes.